everyone, this is Rebecca from the Lab Rat YouTube channel. In this video, we are going to be discussing fixation as a part of our histology lecture series. All right, let's get started. This lecture is on fixation and histology. So what is it? What are we even talking about here? So a fixative is something that alters the tissue by stabilizing the proteins within that tissue so that it is prevented from changing. A fixative must change the soluble contents of the cell into insoluble substances so that those substances are not lost during subsequent processing steps. This change occurs either by chemicals, like using fixative solutions, or via physical means via heat or desiccation and a process called denaturation. Let's talk about this on the next slide. So denaturation causes the protein molecule within the tissue to unfold, and the internal bonds of that protein molecule are disrupted. Now, in the process known as additive fixation, this disruption enables the protein to combine chemically with a fixative molecule, and once that process happens, the protein then becomes insoluble. Now, with non-additive fixatives, like for example, alcohol or acetone, denaturation causes the protein to become less capable of maintaining a relationship with water, and it becomes more reactive. But the fixative molecule doesn't combine with the protein with this non-additive fixative. Recall that we defined fixative as something that alters the tissue by stabilizing the protein so that it is prevented from changing. Now, an older definition of fixative was one that kills, penetrates, and hardens tissues. Penetration is extremely important because adequate penetration of the fixative ensures that the interior of the tissue is fixed, as well as a few exterior cellular layers. Hardening was very important in the early days of histology, since much of the sectioning was performed by hand. Today, with a variety of embedding media, this hardening action is less important than what it once was. So what are the functions of fixatives? One function is to kill the tissue. Another is to help maintain the proper relationship between cells and extracellular substances. And another is to aid in rendering the constituents of the cell insoluble. So let's talk about each of these functions on the following slides here. Okay, so the first function of a fixative is to kill the tissue. This helps to prevent post-mortem activities of decay or putrefaction and autolysis. Now, decomposition occurs at the moment of death, or it starts at the moment of death, or the removal of tissue. So decomposition is caused by two different factors, decay and autolysis. Decay is caused by the breakdown of tissues and cells caused by bacteria. Autolysis, the breakdown of the cells and tissues by enzymes. It's often referred to as self-digestion, so the body's own enzymes are destroying the cells and tissues. Decay caused by an attack of bacteria can be prevented in most fresh tissue specimens by adhering to strict antiseptic techniques. Autolysis cannot be prevented, and it's a very common problem, especially if fixation is delayed in tissues that are rich in enzymes. If a tissue has severe autolysis, it will fail to stain properly. The next function of fixatives is to help maintain the proper relationship between cells and extracellular substances, such as the connective tissue fibers like collagen, reticulin, and elastin, and the amorphous ground substance. This stabilization is important during the subsequent processing steps that might otherwise distort the tissue elements. The fixatives also function to bring out differences in refractive indexes and to increase the visibility of different tissue elements. Now, when I say refractive index, I'm meaning the ratio of the velocity of light in air to the velocity of light in a liquid or solid medium. If air and tissue had the same refractive index, the tissue would be invisible. So enhancing the differences in the refractive indexes of various tissue structures increases the contrast between those structures. Fixatives also help to enhance the stain and aid in rendering the constituents of the cell insoluble. Now, actions of fixatives. Some students may get confused on the difference between functions of fixatives and the actions of fixatives. So functions of fixatives are what their purpose is, so what they do for us in the histopathology laboratory, whereas the action of fixatives is how they do it. So what do they specifically do to that histology specimen? Enzymes, which of course are proteins that are within the tissue sample, are inactivated as a result of the protein stabilizing action of fixatives. This is very important because recall in the last slide that enzymatic action causes the autolysis of the tissues. Tissues that are really rich in enzymes, so for example, the liver, brain, and pancreas, are more susceptible to rapid autolysis in comparison to those tissues that have a predominant amount of connective tissue fibers. Fixatives also kill bacteria and molds, and bacteria and molds can cause the tissue to decay. So if those are killed, that is going to prevent it from decaying. Fixatives also make tissue more receptive to dyes and may act as something called mordants, which serve to link the dye to the tissue. We'll discuss this more in our nuclear and cytoplasmic staining lecture. So fixatives must stabilize the tissue elements so that the effect of any subsequent procedures will be minimized. Now there are both physical and chemical methods that we can stabilize proteins. One of the physical methods is by using heat, and heat will denature and stabilize protein within the tissue. Heating methods were generally not used much in histopathology labs, but with the creation of the microwave oven, it is used more now. We'll talk more about microwave ovens and their use in the histopathology laboratory in our instrumentation lecture. Desiccation is also a physical method for fixing protein, but it is rarely used in the histopathology laboratory. Probably the most frequently used method of this is for the air drying of touch preparations for right staining. Now, the most common method of stabilizing protein in the histopathology laboratory is by using chemicals. The chemical reagents that are used for this process are classified as additive or non-additive and coagulant or non-coagulant. So let's talk about these types of chemicals. Fixatives that are classified as additives are those that chemically link or add themselves to the tissue, and by doing this, change the tissue. When an additive fixative adds it onto a tissue macromolecule, the electrical charge at that site may change. If that charge was once a force that was helping to keep the molecule shape, the change in charge is going to alter the tertiary structure of that protein. Common additive fixatives are mercuric chloride, chromium trioxide, picric acid, formaldehyde, glutaraldehyde, osmium tetroxide, and either zinc sulfate or zinc chloride. So those were additive fixatives. Non-additive fixatives are those that act on tissue without chemically combining with that tissue. So they don't add themselves to the tissue. Primarily, these fixatives dissociate the bound water molecules present within the tissues. This can cause the tissue to shrink and harden if overexposure to the non-additive fixative occurs. Examples of non-additive fixative reagents are organic ones like acetone and alcohols. Coagulant fixatives are those that establish a network of sorts within the tissue that allows solutions to easily penetrate into the interior of the tissue sample. Examples of coagulant fixatives are alcohol, zinc salts, mercuric chloride, chromium trioxide, and picric acid. Non-coagulant fixatives create a gel that makes the penetration of reagents difficult. These include formaldehyde, glutaraldehyde, osmium tetroxide, potassium dichromate, and acetic acid. Now that we have talked about the different types of fixatives, let's discuss the factors that influence fixation. We'll talk about all of these individually, but these are temperature, size, volume ratio, time, choice of fixative reagent, penetration, storage of tissue, pH, and osmolality. So these are factors that affect the quality of the fixation of the tissue sample. So let's talk about those. Okay, so temperature. In general, as temperature increases, the rate of fixation also increases. Also, as temperature increases, the rate of autolysis also increases. For samples that will be evaluated via electron microscopy, that is looked at under the electron microscope, fixation at 0 to 4 degrees Celsius is considered the ideal temperature for it, although some histology laboratories are no longer using cold fixation. Elevated temperatures are being used for fixation in both tissue processors and microwave ovens. In general, an increase in the fixative temperature up to about 45 degrees Celsius is reported to have very little effect on the morphology of tissues. The thickness of the tissue specimen is very important because of its effect on the penetration of the subsequent reagent. If the specimen is a large piece of tissue, such as a segment of colon or small intestine, and is held for an extended period of time without being surgically opened to expose all of that tissue's the layers, the fixative will have a difficulty penetrating through the entire wall to the inner epithelial surface. So this results in the autolysis of the epithelium. To prevent this from happening, large specimens like this should be opened before placed into a fixative reagent. For routine processing schedules, tissue sections should be no more than 3 millimeters thick. A tissue section should never be so thick that it touches both the top and the bottom of the tissue processing cassette. The amount of fixative volume should be at least 15 to 20 times greater than the volume of tissue. Frequently, problems in staining of the tissue are the result of poor fixation due to the use of not enough fixative solution. In an ideal situation, the tissue specimen should be placed in a fixative solution immediately after surgical removal. Also, in an ideal situation, autopsy should be performed immediately after death. This is because the more time that elapses between the interruption of blood supply and the fixation of tissue, the more postmortem changes occur within that tissue. In a couple slides here, I will show you an example of this. Now, the duration of fixation is also important. Adequate time for fixation is necessary so that the tissues will not be distorted by the subsequent processing steps. Formalin fixation should have around 6 to 8 hours of time to act. And of course, the tissue specimen must be left in the fixative for enough time in order for it to be properly fixed. The tissue cannot remain in the fixative solution for an indefinite amount of time. If the tissue is left too long in a fixative, it becomes hardened and staining may be impaired due to this hardening. This is a drawing from Frida Carson's histology book showing a well-preserved small intestine section. The outer layer here, I'll mark it with my little red pointer, is the epithelium, specifically simple columnar epithelium, which lines the small intestinal mucosa. There are occasional goblet cells within the epithelium, so this is what I'm talking about here, these little goblet cells. These are all goblet cells here. These goblet cells form secretions to create a protective mucus layer around the cells. Now, these finger-like projections, here and here, are called villi, which are distinctive of small intestinal tissue. Villi actually help to not only increase the surface area of the small intestine, but also absorb nutrients. Now, in between these villi are crypts of Lieberkühn. All right, so let's get the pointer out here. So these are the crypts here, all right? And these contain Paneth cells, which have secretory granules. So these Paneth cells stain a deep red with eosin stain. Now the tissue underlying the epithelium is called the lamina propria, which is here. So this is the lamina propria, all right? And this contains the connective tissue cells and fibers and blood vessels. Underlying that lamina propria is a layer of smooth muscle called the muscularis mucosa, which is here. Okay. So this is a normal looking section of a small intestine. Here are actual histological sections of the small intestine mucosa. On the left is an excellent preservation sample. So this was from a surgical section that was opened and placed in a fixative immediately upon its removal from the body. We can clearly see all parts of the sample. The epithelium, let me get my pointer out here again. So this is the epithelium. Here are those goblet cells, right? And the Paneth cells, which are here, and those crypts of Lieberkühn, and then the lamina propria, which is here, right? So the lamina propria. Alrighty. So this autolysis was halted in this particular sample because it was immediately fixated upon removal. Now, on the right-hand photograph, this shows a small intestine section that was fixed after a delayed autopsy. As you can see clearly here, autolysis has occurred, only leaving a few glands, and the epithelium is gone from this tissue. Now, most of the goblet cells are also missing. Only the lamina propria can be seen here within this section. Now, these are photos of actual histological sections of lung tissue. The photograph on the left shows a section of lung tissue that was properly fixed before processing. Tissue that isn't well fixed will not process and stain well. The photo on the right-hand side of the slide shows a section of lung tissue that was not completely fixed prior to processing, and it shows poor stabilization of the structure of the tissue. All right, so the method of fixation must be chosen immediately when the tissue sample is acquired. Sometimes the sample actually doesn't even require fixation. Examples of this would be if the tissue is being used for an immunofluorescence study, or if an enzyme profile is needed, which requires the specimen to be frozen without it being put into a fixative solution. Fixatives must be chosen to ensure that a particular tissue element is visible. An example of this would be using Zenker solution as a fixative with muscle cross striations to be stained with Bouin solution when the tissues are to be stained with a trichrome technique. To increase the reaction of the stain, a microscopic section of tissue that has been properly fixed can be treated with another fixative reagent, and this is called post-fixation or mordanting. This is a process used in the Mason trichrome technique, where sections of tissue that were fixed with formalin are then mordanted with Bouin solution before the tissue is stained. It's very important for proper fixation, as some tissue elements won't be visible if the original fixation is improper. Again, fixatives need to fully penetrate the tissues. Factors that determine the minimum length of time the tissue needs to be put in the fixative solution are the rate of penetration of the fixative and the mode of action of the fixative. Most coagulant fixatives give their full effect on tissue as soon as they penetrate the tissue. Formaldehyde is a non-coagulant fixative and penetrates the tissue quickly, but does continue to cross-link proteins for a long time after penetration. Now, in order of decreasing speed of penetration, the fixatives are listed as formaldehyde, acetic acid, mercuric chloride, methyl alcohol, osmium tetroxide, and then picric acid. So the fastest are, of course, formaldehyde, and then it goes all the way down to the slowest, which is picric acid. If a tissue is fixed in a neutral buffered formalin solution, storage is usually not a problem, and it may remain in the solution indefinitely. Appropriate tissue storage will be discussed when we talk about each fixative solution throughout this lecture. The pH of a fixative solution is not very important when the specimen is being viewed using light microscopy. Any pH from 4 to 9 makes little difference when viewing tissues under light microscopy. Now, when using an electron microscope, the pH of a fixative solution becomes much more important. Ultrastructure of tissues is important in electron microscopy, so the pH of the fixative solution should be kept at a pH of 7.2 to 7.4 for these specimens. Osmolality is the last factor that influences the function of fixation. Osmolality is the number of particles in a solution, and like with pH, it doesn't matter as much with light microscopy specimens, but it is important in electron microscopy. Osmolality is measured in milliosmoles. Body fluids have an osmolality of around 340 milliosmoles. When discussing osmolality, the terms isotonic, hypertonic, and hypotonic are used frequently, and we'll discuss these in the next couple of slides. So isotonic is used when describing a fluid into which normal cells can be placed without causing swelling or shrinking of those cells. Saline solution, which is a 0.9% solution of sodium chloride, or 5% glucose solution, are considered isotonic. Isotonic solutions are used in the histology laboratory as a holding solution for tissue samples. Now that we know what an isotonic solution is, let's discuss hypertonic solutions. These are solutions that have a higher number of particles than the cells. So when placed into these solutions, the cells shrink. This occurs because water passes through the semipermeable cell membrane into the solution in an effort to equalize the concentrations on both sides of the membrane. Hypotonic solutions are those that cause the cells to swell. Sodium chloride solutions of less than 0.9% concentration are hypotonic to animal tissue cells. This occurs because the solution has a lower amount of particles than the inside of the cell, and the water outside the cell will move into the cell, causing it to swell. Sometimes the cell swells so much that it will rupture the cell membrane. Again, tissue specimens can be held in an isotonic saline solution, but not for a prolonged period of time. For biopsy specimens, it's better to wrap it in a piece of gauze that has been dampened with the saline solution. It can then be sealed in a plastic container and put on ice for short-term holding. Now, parts of the cell have different reactions with fixatives. The nucleus, which is basically like the brain of the cell, so it controls and regulates all the activities of the cell and contains both DNA and RNA. And it appears that most fixatives do not chemically react with it. For nucleic acids, the fixative's acetic alcohol and carnoy solution are preferred. Formaldehyde doesn't react with nucleic acids until the temperature reaches 45 degrees Celsius for RNA and 65 degrees Celsius for DNA. Coagulant fixatives make the tissue more resilient to sectioning, deparaffinization, and staining, resulting in a more intact appearing nucleus. After fixation in formalin, the nuclei can show a coalescence of their chromatin into strands with clear spaces. This is a phenomenon called nuclear bubbling. This is caused by incomplete fixation. A photo of this is actually on the next slide here. So this is a tumor section that has the artifact called nuclear bubbling. This is frequently associated with formalin fixation. Note that the contrast between the cell nucleus and the cell cytoplasm is good, and there are crisp nuclear membranes demonstrated. Now, proteins have primary, secondary, tertiary, and quaternary structures. The primary structure is the singular chain of amino acids. The secondary structure refers to the hydrogen bonding within those polypeptides, forming alpha helixes and beta plated sheets. The tertiary structure is the overall folding of the protein containing those secondary structures, and the quaternary structure is a multi-subunit complex of all of those 3D structures together. This drawing shows the primary structure, secondary structure, tertiary structure, and quaternary structure of proteins. Sometimes it helps to just see kind of a picture of it rather than just reading words, so I've included it here for you if that helps. So how do fixatives react with proteins? Additive fixatives can cause the 3D shape of proteins to change by changing the electrical charges of their attachment site. Non-additive coagulant fixatives cause proteins to become insoluble, meaning they cannot be dissolved, by altering their tertiary structure. Methanol and ethanol help to preserve the secondary structure of proteins, but greatly affect the tertiary structure of proteins. Lipids, which are fatty compounds, can be preserved by several fixatives. Osmium tetroxide and chromic acid help to fix lipids so that they are maintained throughout the subsequent processing steps. Some carbohydrates are lost during the fixation process. Glycogen, which is the body's storage form of the sugar glucose, can be preserved with fixation. So we have talked about the functions and actions of fixatives, as well as how they interact with the tissue. Now we're going to talk about the actual fixatives. So these are water-based fixatives and fixative ingredients, and these are acetic acid, formaldehyde, glutaraldehyde, mercuric chloride, osmium tetroxide, picric acid, potassium dichromate, and zinc salts. And we're going to talk about all these individually here. So the first one is acetic acid. Concentrated acetic acid is called glacial acetic acid. It penetrates the tissue very rapidly and leaves the tissue very soft. It is classified as a non-coagulant fixative. It doesn't fix or destroy carbohydrates or lipids within the tissue. The major use of acetic acid as a fixative is in the precipitation and preservation of nucleoproteins. So it fixes the nucleus and also precipitates DNA. Acetic acid does tend to swell proteins more than other fixatives, and because of this quality, it can be added to other fixatives to actually counteract the shrinking effect of those other fixative reagents. Acetic acid also lyses or breaks open red blood cells. So any red blood cells that are present in the specimen are not going to be preserved if using acetic acid as a fixative. In terms of storage, acetic acid reagents should be stored at room temperature and away from strong oxidizers, nitric acids, and caustic chemicals. If exposed to acetic acid, it can cause severe burns and has a permissible exposure limit of 10 parts per million. It does need to be transported in a designated acid container and should only be used under a fume hood in the laboratory. Acetic acid should be put in water, never put water into acetic acid when mixing. Now, formaldehyde. So a 37 to 40% solution of formaldehyde is commonly used in the histopathology laboratory. The terms formaldehyde and formalin have been used interchangeably. Now, the textbook written by Frida Carson, the histotechnology textbook, describes formalin as the 37 to 40% solution and formaldehyde as the bottles obtained from manufacturers that these commercial formalin solutions are made from. 10% formalin is the most commonly used fixative. It is classified as a non-coagulant additive fixative. Now, paraformaldehyde, which is a highly polymeric form of formaldehyde, is used in electron microscopy for the preparation of fixative solutions because it yields a pure formaldehyde solution with no additives. It can be added as a powder in concentrated solutions. Formaldehyde reacts primarily with amino acids that contain hydrogen. A major site of reaction is in the amino group on the side chains of amino acids with the formation of methylene bridges. The amino group is positively charged and it attracts the eosin dye during H&E staining. Formaldehyde changes the ability of those positive charges to bind to the eosin stain. Binding of the hematoxylin by negatively charged groups is not effective, so the reaction in the tissue is more basophilic. The greatest binding of formaldehyde is when the pH is between 7.5 to 8. Now, formaldehyde does preserve lipids, but because it does not render the lipids insoluble, there will be a gradual loss of those lipids when the tissue sample is left in formalin for storage. If a tissue is fixed in formalin and then processed for a paraffin embedding, the lipids can be dissolved by using alcohol and xylene. Formaldehyde penetrates tissues very quickly, but fixes slowly because of the need for it to cross-link the proteins within the tissue sample. Complete fixation occurs within 7 days, but about 1/2 of the fixative is taken up by the proteins in the tissues in about 8 hours or so. Formaldehyde causes less shrinkage than any other fixative solution, and it hardens the tissue more than any other fixative solution other than acetone and ethanol fixatives. Formaldehyde can be used in an aqueous solution, or it can be used with the addition of sodium chloride. These solutions do become acidic when exposed to the air. Something called black acid hematin, also called formalin pigment, forms as the result of this. So formalin pigment is a microcrystalline dark brown pigment. It's formed when the formaldehyde aqueous solution becomes acidic and acts on tissues that are rich in blood. It tends to form most frequently when the pH is below 6.0. This can form in any solution that contains formaldehyde that is acidic. You can prevent this formalin pigment from occurring by keeping the pH of the solution near neutral, which is a pH of 7.0. Now, formalin pigment is considered undesirable as it may mask or simulate microorganisms and pathologically important pigments in the stain. It will reduce silver solutions that are used in histology procedures for staining melanin, fungus, reticulin, and spirochetes. So this pigment can be removed by treating the tissue with an alcoholic picric acid or alkaline alcohol. Formalin pigment is birefringent and can be seen using a polarizing microscope. So we use the term birefringent actually in the medical laboratory science profession as well. And what this means is that when something is birefringent, it splits a light wave into 2 waves that are refracted into 2 different directions. You can examine this using a polarizing microscope. Here are photographs of tissue sections showing formalin pigment. The photograph on the left-hand side of the slide shows it present in an area of the kidney that's very rich in blood. On the right-hand side of the slide shows a spleen tissue section with formalin pigment under a polarizing microscope. So viewing sections under a polarizing microscope is a good screening method for the presence of formalin pigment. Formalin solutions are hypertonic, and recall what this means. It means that the solution contains more particles than the cytosol of the cell. So it's important to buffer the solution of formaldehyde in order to keep the pH at a stable level. The more commonly used formalin solutions are 10% aqueous formalin, 10% formalin saline, calcium formalin, formalin ammonium bromide, acetate formalin, 10% neutralized formalin, 10% neutral buffered formalin, modified Millenig formalin, and alcoholic formalin. So now let's talk about all these formalin solutions. So 10% aqueous formalin is created by mixing 100 milliliters of 37 to 40% formaldehyde with 900 milliliters of distilled water. This is a hypotonic solution and can produce formalin pigment. 10% formalin saline is created by mixing 100 ml of that 37 to 40% formaldehyde, 9 grams of sodium chloride, and 900 ml of distilled water. This is an isotonic solution and it can produce formalin pigment. Calcium formalin is created by mixing 100 ml of 37 to 40% formaldehyde, 10 grams of calcium chloride, and 900 milliliters of distilled water. Calcium formalin is recommended for the fixation of phospholipids in tissues. Formalin ammonium bromide is created by mixing 150 milliliters of 37 to 40% formaldehyde, 20 grams of ammonium bromide, and 850 milliliters of distilled water. Formalin ammonium bromide is used only with tissues of the central nervous system. An astrocyte is a type of glial cell that makes up most of the cells present within the human central nervous system. There is something called a Cajal astrocyte procedure, and this fixative solution is used for that. Formalin ammonium bromide is a very acidic solution, and it lyses or breaks open red blood cells. So it causes nuclei to give a direct positive shift reaction due to Fulgen hydrolysis during the fixation process. So this is a type of stain we will discuss later on within our histology lectures. Now, acetate formalin is created by mixing 100 milliliters of that 37 to 40% formaldehyde, 20 grams of sodium acetate, and 900 milliliters of distilled water. This is considered one of the better formalin solutions if you do not want to prepare a buffered reagent. 10% neutralized formalin is prepared by mixing 100 milliliters of 37 to 40% formaldehyde, 900 milliliters of distilled water, and calcium or magnesium carbonate to excess. 10% neutralized formalin has been used as a fixative, though it's no longer recommended because it becomes acidic. 10% neutral buffered formalin is prepared by mixing 100 milliliters of 37 to 40% formaldehyde, 900 milliliters of distilled water, 4 grams of monobasic sodium phosphate, and 6.5 grams of dibasic sodium phosphate. 10% neutral buffered formalin is recommended for routine formalin fixation. It has a pH of around 6.8 and is considered hypotonic. Modified Millenig formalin is created by mixing 100 milliliters of the 37 to 40% formaldehyde, 18.6 grams of monobasic sodium phosphate, 4.2 grams of sodium hydroxide, and 900 milliliters of distilled water. It is considered an isotonic solution and has a pH of around 7.2 to 7.4. It's considered a dual-purpose fixative and allows for electron microscopy on tissues. Alcoholic formalin is prepared by mixing 100 ml of 37 to 40% formaldehyde with 650 ml of absolute ethyl alcohol and 250 ml of distilled water. I'm just realizing actually as I'm doing this lecture that I'm saying mils. I realize that might be a United States thing and I apologize. So mils is, when I say mils, I'm referring to milliliters. Hopefully that doesn't confuse anyone. So this is a compound fixative and is useful on tissue processors because it fixes and dehydrates tissue. Alcoholic formalin should be prepared by taking each component and putting them into a flask for mixing. Safety is a big concern when working with formaldehyde. It's considered a carcinogen, which is a substance that increases the risk for developing cancer. Employees that work with formaldehyde must be monitored. The permissible exposure limit for an 8-hour period is 0.75 ppm. Formaldehyde also has a short-term exposure limit of 2 ppm over a 15-minute period. Glutaraldehyde is similar to formaldehyde, but differs because it is a dialdehyde with an aldehyde group on each end of its molecule. Glutaraldehyde fixes slowly and poorly, so gross tissue sections must be thin in order to use this fixative. It's most frequently used for the fixation of electron microscopy specimens because it preserves the ultrastructure of the tissue better than any other aldehyde fixative. Glutaraldehyde can overharden tissue, so prolonged fixation in this solution is not recommended, usually only 2 hours or less. After fixation with glutaraldehyde, the tissue should be transferred to a buffer solution for holding. Any techniques for staining using Schiff reagent, like the periodic acid Schiff stain, cannot be used on tissues that have been fixed with glutaraldehyde due to the potential of false positive results. And again, talking about that Schiff reagent, we'll talk about it more within our histology lectures. Glutaraldehyde breaks down when exposed to air, dropping its pH. And because of this, the reagent should be prepared directly before use. Glutaraldehyde for use in fixing specimens for electron microscopy should be sealed in small vials before use. In these specimens, glutaraldehyde is used in a cacodiolate or phosphate buffered solution. These can be prepared by mixing 1.9 grams of monobasic sodium phosphate, 0.43 grams of sodium hydroxide, 100 milliliters of distilled water well mixed, and then 18 mils of 25% glutaraldehyde solution. So the glutaraldehyde solution should be handled the same way that formaldehyde is. Mercuric chloride is the next fixative we're going to be discussing. So this is an extremely corrosive chemical, and all contact with mercuric chloride and metallic objects must be prevented. Mercury itself is not used, but is used in compound fixatives because it coagulates proteins and enhances staining by leaving any tissue section receptive to dyes. Mercuric chloride does not react with lipids or carbohydrates, but does inhibit freezing, so it should not be used for frozen sections. This fixative penetrates poorly and can shrink tissue. It is considered an additive fixative, I cannot talk, good grief, and reacts in acidic fixative solutions with the sulfhydryl groups of the amino acid cysteine to form cross-links between protein chains. There is a fixation pigment produced by mercuric chloride. This unfortunately cannot be prevented, but it can be removed. If it's not removed, it shows up as an amorphous brown precipitate in the section. The photo on the left-hand side of this slide shows mercury pigment in a tissue section. Mercury pigment will polarize light, as you can see in the photograph on the right-hand side of the slide. So this is a section of a spleen showing mercury pigment. And recall that formalin pigment can also be seen using a polarized microscope. You cannot tell the difference between mercury pigment and formalin pigment under a polarizer. Mercury pigment can be removed by treating the sections with iodine and then following it up with sodium thiosulfate. The iodine oxidizes the mercury, and the sodium thiosulfate removes the excess iodine from the section. And actually, I probably should show you this here with my pointer. So here is that mercury pigment, and then of course on the polarizer it shows up here. Mercury is a very toxic chemical. Any fixatives that contain mercury must be collected for appropriate disposal, meaning that this is not a chemical that can be put into a sink for disposal. It must be tracked, and careful records must be maintained in the histopathology laboratory. The amount of mercury remaining in the laboratory plus the amount discarded must be equal to the amount of mercury that was received originally in the laboratory. Osmium tetroxide is most commonly used for the fixation of specimens for electron microscopy. It's considered an additive fixative. Specimens for electron microscopy are fixed with aldehyde solution primarily. They are then post-fixed with this osmium tetroxide. This ensures that the lipids in the sample are preserved. Osmium tetroxide can only penetrate through a few cell layers, so specimens must be thin if this is the fixative that's being used. Osmium tetroxide is hazardous and easily vaporizes, so it must be used only under a fume hood. Picric acid has two main uses in histology. It's used both as a fixative and as a stain. This is a coagulant fixative of nucleoproteins, but it does leave DNA soluble. If the specimen needs DNA and RNA stains, picric acid should be avoided as the choice of fixative. Picric acid does not fix lipids or carbohydrates, but leaves the tissue very receptive to dyes and leaves the tissue soft. The only fixative reagent that allows greater final shrinkage than picric acid is actually ethanol. Before processing, picric acid must be washed out of the tissue, usually with 50% alcohol. Care must be taken when using picric acid as it is an explosive compound. As long as the picric acid stays moist, it is safe to use. The jar containing the picric acid must be kept tightly capped for this reason. Potassium dichromate is a non-coagulant unless it is used in an acidic solution. In an acidic solution, it acts as a coagulant fixative. Chromium will render some lipids insoluble, but it doesn't preserve the tissue's lipids as effectively as osmium tetroxide. Potassium dichromate is a fixative that preserves mitochondria but does dissolve DNA. Fixation pigments can occur with the use of potassium dichromate. Potassium dichromate should be removed from the tissues by washing with running water. It is very toxic, both with ingestion or inhalation of the chemical. It is also a carcinogen, which can contribute to the development of cancer and is corrosive to the skin. It must be tracked and collected in the same way that mercury is in the histopathology laboratory. Zinc sulfate can be used in the histopathology laboratory as a replacement for mercury. It is not considered a hazardous waste and can safely be disposed of in a sink. When compared to formalin solutions without zinc, zinc formalin solutions give superior nuclear detail and have better infiltration of paraffin. Other than glutaraldehyde and formaldehyde solutions, most fixative solutions are used in combination with each other. This is a way that disadvantages from one fixative can be counterbalanced by an advantage of another fixative. So this is the list of the most commonly used compound fixatives used in the histopathology laboratory. This is B5, Buin, Gendra, Hollande, Zanker and Helly, Orth, Zamboni, and zinc formalin. And we will discuss all of these now. B5 stock solution consists of 12 grams mercuric chloride, 2.5 grams of sodium acetate, and 200 milliliters of distilled water. The working B5 solution contains 20 milliliters of the B5 stock solution and 2 milliliters of 37 to 40% formaldehyde solution. B5 is used as a fixative for hematopoietic and lymphoreticular tissue because it creates a beautiful nuclear detail in the section. After fixation with B5, the tissue does not need to be washed, but it does need to be treated for the removal of mercury pigment. Recall when we were discussing mercury pigment earlier in this lecture that it can be removed by using iodine and then following it up with sodium thiosulfate. Tissue should be placed in the B5 fixative for 4 to 8 hours. After fixation, the tissue should be placed in a wet tissue with 70% alcohol. Because this solution contains formaldehyde and mercury, all precautions that are applicable with formaldehyde and mercury should be used. The photo on the left-hand side of this slide shows a lymphoma section that has been fixed with B5 solution. Note how it has crisp and clear chromatin patterns of the nuclei. Now the photo on the right-hand side of this slide shows a lymphoma section from the same tumor as the one on the left-hand side, except that it has been fixed in 10% neutral buffered formalin instead of that B5 solution. So note the difference in the fixation of the nuclei between these two sections. Buin solution is prepared by combining 750 milliliters of saturated aqueous picric acid solution, 250 milliliters of 37 to 40% formaldehyde, and 50 milliliters of glacial acetic acid. Because the Buin solution contains acetic acid, red blood cells are lysed or broken open with the use of this fixative solution. It dissolves iron and calcium deposits, but may produce formalin pigment. Buin solution is a great choice for trichrome stains and for the preservation of tissue that is soft and delicate. Tissue that is fixed with Buin solution easily sections and causes a yellow color, which must be removed from the tissue by washing with 50 to 70% alcohol. The remaining wet tissue should be stored in 70 to 80% alcohol because it cannot remain in the Buin solution for a long period of time. Maximum time in the Buin solution should be less than 24 hours long. Now, since it does contain formaldehyde, all regulations for the use of formaldehyde should be used when using this solution. It is a great fixative for GI tract specimens and for those of the endocrine system. It cannot be used for tissues that are going to be viewed with electron microscopy or for those where nucleic acids are to be examined. Gendre solution is created by mixing 800 milliliters of 95% alcohol saturated with picric acid, 150 milliliters of the 37 to 40% formaldehyde, and 50 milliliters of glacial acetic acid. You may notice that this sounds similar to the chemicals for the Buin solution. That makes sense because this is an alcoholic Buin solution, and it's great for preserving carbohydrates, most specifically glycogen, which is the storage form of glucose within humans and animals. After fixation, the tissue should be washed with 80% alcohol. Hollandaise solution is prepared by mixing 25 grams of copper acetate, 40 grams of picric acid, 100 milliliters of the 37 to 40% formaldehyde, 15 milliliters of acetic acid, and 1,000 milliliters of distilled water. This is a modified form of the Buin solution and will decalcify bone. It can be used for the fixation of GI tract specimens. The cupric acetate in this solution stabilizes the membranes of the red blood cells. This allows for less lysis of the red blood cells than in regular Buin solution. The tissue must be washed after fixation with this solution. Zenker and Heli-Stock solution is produced by combining 50 grams mercuric chloride, 25 grams potassium dichromate, 10 grams of sodium sulfate, and 1,000 milliliters of distilled water. The Zenker working solution is produced by combining 95 milliliters of the Zenker Heli-Stock solution with 5 milliliters of glacial acetic acid. The Heli working solution is created by combining 95 milliliters of the Zenker Heli solution with 5 milliliters of 37 to 40% formaldehyde solution. If tissues are treated with any of these fixatives, they must be treated to remove mercury pigment. Because the Heli working solution contains formaldehyde, it can also produce formalin pigment. Zenker solution will lyse red blood cells in a tissue sample, so break up red blood cells, whereas the Heli solution will not. The fact that Zenker solution contains acetic acid means that it will be a better nuclear fixative. Tissue cannot remain in any of these solutions longer than 24 hours. If so, the tissue will be over-hardened. After fixation, the tissue should be washed in running water and stored in 70 to 80% alcohol. Those with mercury and formaldehyde must be handled in accordance with all hazardous waste regulations. Orth solution is made by mixing 2.5 grams of potassium dichromate, 1 gram of sodium sulfate, 100 milliliters of distilled water, and just before using 10 milliliters of the 37 to 40% formaldehyde is added. Or solution is not a good general purpose fixative, but is preferred for the chromate living granules in the cytoplasm of cells from the adrenal medulla. This is the inner part of the adrenal gland, which controls the hormones epinephrine and norepinephrine. The granules are important in the diagnosis of something called pheochromocytoma. This is a neuroendocrine tumor in the adrenal gland that grows from the chromaffin cells. Now the tissue must be washed after it is fixed with ORF solution, and the remaining wet tissue should be stored in 70 to 80% alcohol. Zamboni solution, which is also called buffered picric acid formaldehyde, or PAF, is prepared by mixing 20 grams of paraformaldehyde with 150 milliliters of saturated aqueous picric acid. The solution is heated to 60 degrees Celsius to help dissociate the paraformaldehyde. Then 2.52% aqueous sodium hydroxide is added to make the solution more alkaline. The solution is then filtered and allowed to cool. Once it's cooled, it must be diluted with 1,000 milliliters of phosphate buffer. Zamboni solution should have a pH of 7.3. It is a good purpose fixative and is preferred by some to be the best fixative for electron microscopy. Alcoholic zinc formalin is prepared by mixing 4.5 grams of zinc chloride with 1,600 milliliters of deionized water. This mixture is stirred until fully dissolved. Then 2,000 milliliters of 99% isopropyl alcohol is added, along with 400 milliliters of the 37 to 40% formaldehyde solution. This is used as a post-fixative solution with neutral buffered formalin solution. Nuclear detail of the specimen is an improvement with alcoholic zinc formalin than with using regular formalin fixation. Alcoholic solutions are recommended if 6 to 8 hours cannot be set aside for fixation time. Lastly, unbuffered aqueous zinc formalin can be prepared by mixing 1 gram zinc sulfate with 900 milliliters of deionized water. The solution is stirred until completely dissolved. At that point, 100 milliliters of the 37 to 40% formaldehyde is added. Formalin pigment can be caused by this fixative. 4 to 6 hours of fixation is required for biopsy tissues fixed with unbuffered aqueous zinc formalin solution, 6 to 8 hours for other tissues. Non-aqueous fixative ingredients are acetone, alcohol, and carnoy solution. These are non-additive coagulating fixatives. It's important to use caution as these reagents are flammable and must be stored properly in fireproof cabinets. These fixatives should only be used when the desired components of tissues are destroyed or dissolved by the aqueous fixatives because they do overharden the tissue. Acetone is a non-additive coagulant used when the demonstration of enzymes is needed on tissue that is to be processed for paraffin embedding. It can also be utilized for brain tissue needed for rabies diagnosis. It is commonly used on frozen sections of tissue that needed to be stained for antigens on the cell surface using immunohistochemical techniques. Acetone is very quick as a fixative and causes extreme shrinkage of the tissue. It is narcotic in high concentration, and contact with the skin can cause dermatitis. It is moderately toxic if ingested as well. Ethyl and methyl alcohols can be both used for the purpose of fixation. Alcohols are considered a non-additive fixative. Methyl alcohol is used as a fixative for touch preps and blood smears, whereas ethyl alcohol is used as a fixative for water-soluble components of tissues, including glycogen, which is the storage form of the sugar glucose, and urate crystals in a disorder called gout. Ethyl alcohol preserves most of all pigments, dissolves fat in the tissue, and both overhardens and shrinks the tissue specimen. When combined with formalin, the shrinkage effect is lessened. Carnoy solution is prepared by mixing 60 milliliters of absolute ethyl alcohol, 30 milliliters of chloroform, and 10 milliliters of glacial acetic acid. Carnoy solution lyses or breaks apart red blood cells. It's a very quick-acting fixative, preserves glycogen within the tissue, and preserves the nucleus well. It does cause shrinkage and hardening of the tissue. Tissue should not be in this solution for longer than 4 hours. After fixation, tissues should be processed through 95% alcohols, absolute alcohol, and xylene as usual. Carnoy solution should only be used for the preservation of special tissue components that are lost in the routine fixation process. All right, well, hopefully you're all still with me. I know this is a long-winded lecture here. We're almost done. You've almost made it here. So next we're going to be discussing how specimens are transported. If tissue that is unfixed is only going a short distance, the best method for transport is to place it in saline dampened gauze and then seal it in a container and place that container on ice. Now, if the specimen is going to be held for several days, then the use of a missile transport medium is indicated. This transport medium can be bought commercially. It is important to keep the pH of the transport medium between a pH of 7 to 7.2. Now, we've talked throughout this lecture about pigments caused by fixation. Formalin and malarial pigments must be removed before any processing happens on the tissue. This is done by one of two types of solutions. The first option is absolute alcohol that is saturated with picric acid for up to 3 hours. After this, the sections must be washed well with water. The second option is 70% alcohol that contains 3 milliliters of ammonium hydroxide from 30 minutes up to 3 hours. After this, the tissue must be washed and rinsed in 1% acetic acid, then washed again. Now, mercury pigments must be removed by treating the deparaffinized and hydrated sections with gram or Lugol iodine for 10 minutes. After this 10-minute period, the sections must be washed in water, then placed in a 5% solution of sodium thiosulfate for 3 minutes. And after this 3-minute period, the sections must be washed for 10 minutes and then stained if desired. Alright, so that wraps up our lecture on fixation in the histology laboratory. If this video helped you out, go ahead and give it a like. And if you have any questions, feel free to leave them in the comments below. I will try to answer as many as I can. Alright, until next time.